Hello everyone, welcome to the session. Today we are going to discuss a very important topic called Basic Laboratory Safety and Risk Assessment. In every laboratory, safety is the first priority because even a small mistake can lead to serious accidents, contamination, or injury. So, in this session, we will understand how laboratories maintain safety and how risk assessment helps prevent hazards. By the end of the session, you will understand the importance of laboratory safety, different types of laboratory hazards, the concept of risk assessment, emergency preparedness, and the importance of PPE and safety laboratory practices. These concepts are essential for every student, researcher, and laboratory professional. First, let us understand what laboratory safety means. Laboratory safety includes all the practices followed to protect laboratory personnel, equipment, samples, and the working environment. Safety procedures help prevent accidents and ensure experiments are performed in a controlled and professional manner. Without safety, no laboratory can function properly. Now, let us discuss why laboratory safety is important. In laboratories, people work with chemicals, biological materials, electrical equipment, and sometimes hazardous substances. If safety procedures are ignored, accidents may occur, contamination may spread, and laboratory work may stop completely. Good laboratory safety improves productivity, reliability, and confidence in scientific work. There are different types of hazards present in laboratories. Chemical hazards include toxic chemicals, flammable substances, and corrosive reagents. Biological hazards include microorganisms, infectious agents, and biological samples. Physical hazards include broken glassware, fire risk, and electrical equipment accidents. Understanding hazards is the first step towards laboratory safety. Now, let us discuss personal protective equipment, commonly called PPE. PPE acts as a protective barrier between laboratory hazards and the person working in the lab. Common PPE includes lab coats, gloves, face mask, safety goggles, and closed footwear. Wearing proper PPE reduces exposure to chemicals, infections, and injuries. Safe laboratory practices are daily habits that help maintain safety. For example, following SOPs properly, labeling chemicals correctly, maintaining cleanliness, avoiding food and drinks inside laboratories, and reporting accidents immediately. These practices may look simple, but they greatly reduce laboratory risk. Now, let us move to risk assessment. Risk assessment means identifying possible dangers before starting laboratory work. It helps laboratories recognize hazards, evaluate possible risks, and implement control measures before accidents happen. In simple words, risk assessment helps laboratories stay prepared instead of reacting after problem occurs. Risk assessment generally follows four important steps. First, hazards are identified. Second, the seriousness and likelihood of the risk are evaluated. Third, control measures are implemented to reduce the risk. And finally, safety procedures are continuously monitored and reviewed. Risk assessment is not a one time activity, it is a continuous process. After identifying risk, laboratories apply control measures. These may include engineering controls like fume hoods and biosafety cabinets, administrative controls such as training and SOPs, and personal protective through proper PPE usage. The main goal is to reduce risk as much as possible before accident occurs. That is why prevention is always better than correction. Chemical safety is very important in laboratories because improper handling can cause burns, poisoning, fire, or explosion. Laboratory personnel should read labels carefully, understand MSDS information, store chemicals properly, avoid direct exposure, and follow proper disposal methods. Careful chemical handling protects both personal and laboratory environments. Biological safety, also called biosafety, focuses on handling biological materials safely. Laboratories working with microorganisms and biological samples must follow strict precautions. Important practices include using aseptic techniques, disinfecting work areas, safe disposal of biohazardous waste, and using biosafety cabinets when necessary. Proper biosafety reduces contamination and infection risk. Even after taking precautions, emergencies can still happen. That is why laboratories should always be prepared for emergencies. Essential emergency systems include fire extinguishers, first aid kits, emergency exits, spill management procedures, and eye wash stations. Quick response during emergencies can minimize damage and protect lives. Safety culture means creating an environment where every person takes responsibility for safety. A strong safety culture includes awareness, discipline, teamwork, regular training, and reporting unsafe conditions immediately. Safety should not depend only on rules, it should become a habit followed by everyone in the laboratory. Risk assessment provides many benefits to laboratories. It helps reduce accidents, improve laboratory efficiency, protect personnel and equipment, support regulatory compliance, and improve confidence in laboratory operations. A laboratory with proper risk assessment systems can work more safely and effectively. So, let us summarize today's session. Today, we discussed basics of laboratory safety, common laboratory hazards, PPE, safety laboratory practices, risk assessment procedures, chemical and biological safety, emergency preparedness, and safety culture. Overall, laboratory safety is essential for quality scientific work and protection of personnel. Thank you everyone for watching this session. I hope this video helped you understand the basics of laboratory safety and risk assessment. If you have any questions, feel free to mention them in the comment section. Thank you.